It's my great pleasure to introduce the next speaker, Dr. Nadia Rosenthal. She is the director of the Institute for Regenerative Medicine in Melbourne, Australia. And Nadia had an interesting professional journey because she worked all over the world, including in Italy for 10 years, UK, France, and now she's Australia. And I just heard that she will come back to the States soon. But I was always impressed by the faithfulness that Nadia had to her science and her mission, which was really to understand the biology of muscle and heart stem cells, to understand the signals and the growth factor that are fundamental for the expansion of these cells, with the ultimate goal to bring therapy to patients with incurable diseases. So it's really an honor for me to introduce Nadia, and I thank you for being here from Australia. Thank you.
Well, good morning. It's wonderful to be here. The weather's almost as nice as Melbourne. And I'd like to spend the next few minutes giving you a bit of a taste of what happens behind the scenes. We've heard about the extraordinary advances of clinical practice and trials in the gene and stem cell space from Martin. And what I do is I'm just a nerd, so I work behind the scenes at the bench. And I want to tell you about sort of the way we're going, thinking about the way in which the problems we've heard about from several of the speakers in childhood diseases where regeneration is absolutely critical to the healthy functioning of the adult play out in the lab. So I've been obsessed all my life with this issue. Why is it that these other organisms, such as planaria, little flatworms, or salamanders, or any of these wonderful marine creatures like sea urchins that you find in the pools, why can they regenerate and we can't? It's not like we're missing huge chunks of genomic information. If anything, we have more. And yet we seem to have forfeited this capacity to regenerate for some unknown reason. So what genetic programs have we lost? We really don't know that answer, but I'm going to explore a few of the ideas that we are coming up with in the next few minutes. One of the things that's very clear is that we aren't that bad at regenerating. So we couldn't have lost that much because as fetuses, we're known to be very good regenerators. Mammals can regenerate large chunks of their body as fetuses, and indeed, even neonates, as clinicians know, have an extraordinary capacity for repair. So we think that the basic tools and tricks of the body are still there, but they somehow get muddled up as we get older. And it doesn't take long because, as you know, even by the time you're an adolescent, you're not as good at regenerating. So the advent of regenerative medicine is a new area that's really just about a decade old, seeks to identify the mechanisms whereby we've lost that capacity to replace and restore our tissues, and to ask very simple questions like why we can't grow back that limb, or why we can't restore muscular dystrophic tissue that has been lost due to a basic genetic defect. So I'm just going to give you a bit of a tour through some of the areas that we are thinking about and the way we're approaching this problem at the coalface. So, of course, stem cells are a great feature of regenerative medicine. In fact, most people don't think regenerative medicine is anything other than stem cells. In fact, when I decided I was going to try to call this institute in Australia the Regenerative Medicine Institute, one of the deans said to me, "But isn't that just stem cells?" So I had to explain to him that one is a tool and the other is a discipline. So what we're going to talk about here is a tool, and they're very important tools. And as you know, a stem cell is essentially a cell that can regenerate itself over and over again, as well as become many different kinds of cells. And of course, the ultimate stem cell is the pluripotent egg, our fertilized egg, which makes a human being or a mouse every time. However, there are obviously ways in which we can think about using stem cells in a slightly less dramatic way, but it's just as important clinically, and that is to treat a number of diseases. And there is an enormous amount of promise out there. And indeed, these are some of the areas where people have considered using stem cells to replace tissues in a regenerative medicine scenario. So I'm going to go over a little bit about what stem cells today are being used for. And we all have heard of embryonic stem cells. They're the famous ones that can make any tissue in the body. There's a lot of controversy because of their origin in human embryos for humans. But they do perform these extraordinary feats of becoming all these different tissues, and we can use them to understand development. And as a result, they are an extraordinarily important tool in developmental biology. And indeed, very recently, although I never thought I would say this, I had a slide actually in this talk that said it'll be decades before we use embryonic stem cells in the clinic. So I had to take it out because we're already doing it. And this is a case in point. Very recently, several groups in Japan and in the UK have used embryonic stem cells to imply to the degenerating back of your eye, which creates a scar seen here in this white area. And by applying this to a very localized part of your body, namely an eye, which is sort of immunoprivileged, a little bit isolated from the rest of your body, so a really great place to try this out. There appears to be, at least safety-wise, no problem with putting embryonic stem cells in there. They become the right thing, and so far they've been quite impressive in terms of the capacity to restore vision. So this is amazing. I'm very excited that this has happened so quickly. Now, more frequently you hear about adult stem cells, and adult stem cells are simply the cells within your own body, once you have been born and you have all these different tissues, that are responsible for renewal of the tissues that you already have. So, for instance, the reason that we don't all look like cauliflowers by this point in our life is because we're continuously regenerating things in the right way. So our skin is regenerating skin, our intestines are regenerating intestines, and our blood is making new blood all the time. And some of our body is very good at regenerating, our liver, thank God, and other parts of our body are not so good, such as our brain, as we heard about before, and our heart. So it's not clear just exactly how these different tissue-restricted cells work, and there's been a lot of confusion about just how pluripotent they are. So can you take a cell out of a muscle and make it into a blood cell, or the opposite? Can you take a brain cell and make it into something else? And depending on who you talk to, there's a variety of opinion, the most extreme of which is that there are certain cells that become cells for that particular tissue and no other tissue. And then other people feel there's more plasticity, and we're still, I think the jury's out. There is one case in point, however, where there is no question of pluripotency, and that is in the blood. And we're sitting in the home of Irv Weissman, who is really the father of this whole field. We know that indeed we've been using stem cell therapy ever since bone marrow transplants were discovered and started to be used in some detail, and we've been just hearing about some of the extraordinary capacity for bone marrow and cells of the immune system and the hematopoietic system to form a really important clinical tool for treating blood diseases. And so in the case of hematopoietic stem cell, it can become every single cell type of the blood in the right condition. And this is, of course, the prime example of an adult stem cell that is pluripotent. So, as a result, stem cells in the adult have become very exciting, and there's rumor of cells that are stem cells in capacity to rebuild your body from something as unlikely as your adipose tissue. There are other cases where people have used fibroblasts. There's a lot of hype, and indeed, if you go to Mexico, you can have it done on the beach, apparently, which really always makes me wonder just exactly what's going on at these places. And I think, as we all know, there's a considerable amount of concern about the safety of some of these less controlled institutes where this is going on. But it's an area that I think is really going to gain in importance. So one of the things that happens when adult stem cells are created is that if they are not from yourself, they could create an immune response. And one of the ways around this is, and one of the ways around using embryonic stem cells, is the new induced pluripotent stem cell. And this is just by way of review, a very well established, but very rapidly established new technology for which several people won the Nobel Prize a couple of years ago, in which a run-of-the-mill fibroblast out of your body, literally out of the side of your cheek or anywhere else, can be reprogrammed to recapitulate the entire developmental process of your own embryogenesis, such that you produce a stem cell that can become many other kinds of cells in your body. And of course, because it's your own cell, the genes are your own genes, and the proteins are your own proteins, there'll be no immune response, and they, in theory, could be the perfect precision medicine tool. And indeed, here are two examples. You see at the top there's an example of 4 different genes that are being introduced into fibroblasts and producing a cell that is pluripotent and therefore can become any kind of tissue. And at the bottom, more recent advances allow you to very transiently introduce these factors, which otherwise might be quite dangerous because, of course, they're important for embryogenesis, but they could also be cancer-causing. So there's a lot of technology around this. It's a very exciting field, and we're using it as scientists to know much more about the developmental process that takes us from the fertilized egg to the baby. Here's just a few examples of the idea behind the pluripotent revolution, where you take a patient's stem cell, turn it into the cell they're missing, and reintroduce it into the body. You can also use these cells for pathological studies, toxicology studies, and also, of course, for drug discovery. And indeed, there have been some real advances in this field. Just recently, there was a beautiful study looking at stem cells that were derived from the fibroblasts of patients that had dementia, looking at the gene mutations in those patients and correcting the gene mutation in the patient using gene editing technology in the cell. And then looking to see how those cells were capable of differentiating back into neurons. And although there's a lot of hurdles to still be overcome, the concept is really there, the proof of principle is there. So what are the limitations of stem cell therapy? Well, one possible limitation is how do you get from a cell that is pluripotent to a cell that's differentiated, and how far do you get? And in other words, if you get a neuron, is it a neuron that will really do what you want it to do? Will it learn how to play the piano? And it's not clear that we're really in possession of the tools that will allow us to make those final decisions about really very, very refined differentiation. And specifically, when you transplant a cell into a body without that information, I always liken it to having someone parachute out of a helicopter into the Gobi Desert with no cell phone or any other instruments of sustained viability like water. And how would you expect someone in a business suit to survive there? And in some ways, I think of stem cells that way. When you introduce them into the body, they are completely out of context, and for the most part, they die. So really, the functional integration of stem cells back into the body is something that is an extraordinary hurdle that really isn't talked about enough. So one reason why I think this is a problem is because we don't really understand this very, very fine balance between the inflammatory response that occurs in injury and in degenerative disease and the cell replacement that must eventually supplant that inflammation. So what normally happens is in a very healthy kid, an injury produces a rapid response of the immune system to produce the inflammation necessary to guard against infection and to clean up the mess. But then very quickly after that, the inflammatory response is quelled and cell replacement starts, be that stem cell or other, so that scarring is avoided and repair is favored. And that tipping of the balance is something that we don't know much about. We know that it can be done very easily in the fetus. We know that that balance is definitely skewed as we get older. In the wrong direction. So in my institute in Australia, we've taken the attitude that, again, because we know that there are organisms out there that can do this effectively as adults and don't scar, perhaps we can learn from them. And here are just a few of them. There's zebrafish there on the left, and my favorite new organism, the axolotl, in the middle, and a little mouse on the right. So the idea would be to learning from the variation within the natural animal kingdom will allow us to apply those basic principles in the clinic. So here, as I said, is my favorite new organism. It's always a winner winner because it looks like something that comes from Mars, or definitely from Steven Spielberg. But it is actually a real live organism. It's an aquatic salamander. They live in Mexico. I think there's the second allusion to Mexico. I must be heading for Mexico. So these animals are scar-free. They will heal just about anything you cut off them other than their head. And they are extraordinarily robust in their capacity throughout life to do this. So the question is, why do they die? And that's an interesting question. I'll get to that in a minute. We think that maybe their immune system is different because they never scar, and we know that inflammation is a function of the immune system, and it seems like it's quite different. And it allows these animals to reconfigure their entire limb or tail or heart or jaw, recapitulating the entire developmental sequence within a very short time and making something much bigger than they made at the beginning, because of course they started off with eeny-weeny tiny little arms when they were eeny-weeny tiny little salamanders. Now they've got to grow back a whole adult one, and they do it in the same amount of time. So one asks, why would these animals do this? I mean, why would they be able to grow these limbs back? And the answer is because they eat each other up all the time when they're stressed or hungry or crowded, sort of like academics, and they continue to sort of munch on each other. So the way I got this through the ethics committee down in Australia, they have a very strict ethics committee, and I said, I'm going to cut off the limbs of salamanders, and they said, I don't think this sounds very good. And I said, I'm doing it with anesthesia, unlike the real world. So they are very, very good at regenerating, I think, because of just evolutionary necessity. And what is different? Well, if you look at the way immune cells arrive at an injury during the repair process, there's a stately progression, and this is a picture from the way it looks in most mammals. You have these neutrophils, which are pro-inflammatory cells, and then macrophages, which are sort of phagocytic and eat up other cells that are diseased or dead. There are T cells, which are part of the adaptive immune system. Everybody's on board, but it usually takes about 5 days for this all to happen during an effective regenerative response in a mouse or a child. The axolotls do it in 24 hours. There's a giant cocktail party. Everybody comes together and they arrive at the scene within hours. And we don't know what that means, but we suspect that there's an acceleration of the entire inflammatory response. It's very important for the effective regenerative process to continue. So we did a little experiment. I'm not going to give you too much more data on at all, but I'm just going to tell you a couple of experiments. Here we eliminated one of the key parts of the immune response, which is macrophages. And we did this by essentially having them self-destruct. It's not important. To ask the simple question, do macrophages, they're looking at the purple people eater there, do they actually play an important role in regeneration, or are they just coming along for the ride as an inflammatory cell? We know that macrophages do a number of things, including protect against infection, but interestingly, during cancer metastasis, they also chaperone cancer cells around the body and get them to extravasate out of the circulatory system and move into new areas. And it's that cooperation, that very, very nefarious cooperation, which actually allows for metastasis to continue. So we wondered whether in the regenerative context, this same kind of chaperone approach was going on, because often cancer and regeneration are two sides of the same coin. And so what we did was to cut off the salamander's limb, either with or without macrophages in its system. And I think this is a self-explanatory slide, because you can see that the new limb that is now grown out from that cleavage site, which is the dotted line where we cut off the limb, it's just magic. It grows out and becomes a fully functional and completely recapitulated structure. But without macrophages, even for a few days, right at the beginning of the process of healing, even if they come back later, which they do very quickly, the poor little guy never heals, because there's this massive scar there, and essentially there's never an outgrowth. And the animal remains completely stumped for the rest of its life, unless you cut off a little bit of that scar and you reactivate the system, in which case, boom, the thing starts up again. So clearly you haven't changed anything in the long run. You've just blocked a system which is ready to go, and for some reason needed macrophages to do it. So here's my dream, without, obviously total hype here, but this is one of the happiest Olympic stars from Australia, Paralympic star from the 2012 Olympics in London, and wouldn't it be nice if we could find a way to change her macrophage response and grow back that limb? Just a joke. Anyway, macrophages do a number of things, and so the question is, what are they doing, and what does this have to do with regenerative medicine? Well, it turns out that macrophages are involved in angiogenesis, fibrosis, immune modulation, cell survival, as well as their cleanup mode that we think of. And in each case, they make a particular factor, which I'm particularly interested in, called insulin-like growth factor 1. And so every single one of these functions requires IGF-1. So we wanted to know whether there was something magical about this particular growth factor that macrophages make. And we know that it's important for growth in the body during embryogenesis and early life in childhood, but that later on it's made exclusively by the liver, and it's essentially there for endocrine functions and has some overlap with the insulin. So we know from work we've been working on for a long time when I started in Rome, making mice that overexpressed IGF-1 in their skeletal muscles, and they get very, very muscular. They're sort of Schwarzenegger mice. I was famous for 15 minutes there when it hit the news. But more importantly, when you injure these mice, they regenerate much, much more fast, much more quickly, and they do it in a way that essentially resolves the inflammation much more quickly, just like the salamander. So by simply adding one factor, one additional factor, we've managed to change the course of the traumatic injury to muscle. And here you see an experiment we did a long time ago showing that indeed muscular dystrophy, which is in this case modeled in the mouse, in the MDX mouse, could be countered by the presence of IGF-1. So it looked good, and so we wondered where it was coming from. And fast forward 10 years, Joe Tonkin in the lab has done an extensive study to show that during the injury response, macrophages arrive at the scene and they absolutely flood the area with IGF-1. And if you use genetic tricks to reduce the IGF-1 only in the macrophages, you essentially end up with muscle that cannot regenerate. So that means that almost all of the IGF-1 that's normally a spike during the regenerative response is absolutely critical, and it's coming from macrophages. And without it, you can't regenerate. So the possibility then remains, and I'm not going to go into the details of exactly what we're doing, but we're obviously involved in therapies and trials involving IGF-1. The idea would be to use the immune cells as a kind of Trojan horse to bring these naturally occurring molecules in and perhaps increase the capacity for regeneration by allowing the immune system to do the job. And the idea would be that a pluripotent stem cell that you could, now you know, get out of a patient could be modulated to produce therapeutic factors. And we have other data to suggest that the adaptive immune system, the T lymphocytes, also make IGF-1 for the same purpose. Okay, so finally, just one more story. The heart that can't heal. We know that heart attacks in adults are very, very likely to be fatal, or at least the morbidity associated with a heart attack resulting in heart failure is a serious problem in our society. The reason for this is that the heart, unlike the salamander or our skin or our blood, simply doesn't seem to be able to regenerate at all once it's formed. However, what's interesting is that very recently it's been realized that neonatal mice can regenerate their hearts just as well as a salamander can, and that this is something that gets lost within the first 7 days after birth. Now remember, mice get born at a much more premature stage than a human, so we would be looking at late fetal life in the human context. But for a mouse that's born at this stage, they are literally fully regenerative. And more interestingly than that, we've discovered very recently that if you look across different mouse strains, they regenerate completely differently. And this was something that Matt Gilman brought up earlier, this idea of the context of the individual. So it turns out there are some individual mice that regenerate almost as well as the mice where we make IGF-1 overexpression, and others that regenerate much, much worse than our normal mouse models. And so we're now beginning to use that as a genetic tool to map out the complex traits associated with regeneration in an unbiased way. So if we look at patients, we know that certain patients recover from heart attack much more easily than others. And the question in my lab now is, what is it about this variability within patients that we can model in an animal model such as the mouse? And more importantly, can we use those regenerative variations within the species to develop new tools to treat the disease? And specifically, we've used IGF-1 in this case to overexpress in the heart and shown that indeed just overexpressing IGF-1 alone is enough to literally recapitulate the more regenerative capacity of a neonate. Not surprisingly, the neonate's making a lot more IGF-1. I'm not saying this is the only factor important for regeneration, but I'm saying that if you use it as a sort of a hook to pull out the rest of the network, we may be able to understand the complexity of the regenerative response. So just in conclusion, I want to make a couple of points. One is that there's not going to be one therapy that's going to do the job for a regenerative medicine. It's going to require stem cells, genes, and probably scaffold as well. So that gives that parachuter something to come down in, maybe a bubble of other things that would help survival. A camel, someone once said to me, just give them a camel. So maybe that's what the scaffold will be. And indeed, when we look at some of the more recent ways in which stem cells have been used in heart repair, this is a very beautiful study out of Minneapolis just published a few weeks ago, where a patch of essentially biomaterial was infused with little spheres containing slow-release IGF-1, and that patch was capable of helping with the healing of an infarcted heart, but it was not necessarily any better than putting in induced pluripotent stem cells that had been turned into various kinds of heart cells. But when you put them together, you got a really good response. Namely, if you had the IGF-1 in that patch overlaying the stem cells that you had injected into the heart, then you saw some really much better recapitulation of the regenerative tissue. So that's just one example of how I think the whole thing is going to work. So in summary, this is what I've told you. Stem cell-based therapies are promising. We need to know a lot more. Early resolution of inflammation is a major determinant and is very important. And the future advances will involve thinking about how we treat individuals rather than groups. Looking at the way in which immune systems, the individual variation in people's immune systems may play a great role in allowing our stem cell-mediated therapies and gene therapies to work. And then finally, the combinations of all of these together is probably going to be what it needs to regenerate a body, just like it does in real life. So here are just some of the people that have done the work in the lab. I don't have time to go into all the wonderful postdocs and students that have taken this forward over the years, and I'd like to also thank my sponsors who have made all this work possible. Thanks for your attention.